Hey everybody, thanks for being here today. I hope your week is going really well. Today I'm going to be telling you a little bit about how muscles contract. Now, the only thing that muscle tissue can do is contract. Muscles cannot push forcefully, they can only pull with force. And so when you have a muscle acting on a joint and it pulls, it's going to pull bones together. Now, if you want this joint to be forcefully moved the opposite direction, you need an antagonist, another muscle here that's going to be pulling on this other part of the bone. But it's still, in both cases, the muscles are only pulling. What we'll cover today is at the microscopic level, what the muscle cells are doing to provide that force, to provide that pull. So this is going to be basic muscle physiology. So this is going to be all about basic muscle physiology. The word physiology means at the microscopic level how the body works or how parts of the body work. So really the question we're going to be answering is, how do muscles contract? Muscle cells are specialized. Their cytoskeleton is arranged very uniquely. So actin was part of the cytoskeleton for all cells, but the actin provides a very specific function for contraction in a muscle cell. Actin molecules are globular proteins, but they all fit together into an actin filament. The actin has a small part of it that is a binding site for another protein called myosin. And what that means, the binding site means that those two things are attracted. So this part of the actin molecule is attracted to myosin molecules. So a myosin molecule has a long part and then a head that can swivel. And at the end of the head, it has an actin binding site. And so when they're allowed to, when these both are exposed, they would bind together. So again, the actin forms filaments with all of the binding sites kind of lined up. The myosin filament has a bunch of these with their heads and then another one and a head. So I've represented 4 of them here, 4 of these, and then 4 in the opposite direction. When you look at a muscle cell, you'll see these, and these are called thick filaments, and the actin filaments are called thin filaments. This diagram is of something called a sarcomere. A sarcomere is really the distance between these two things called Z-lines. These lines are representing the actin filaments, so an actin filament here, 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 as well as these. All of these thin filaments are actin filaments. All of these thick filaments are myosin, represented here. These that are connecting, they're called Z-lines, and they are made of a different type of actin, plus other proteins like titin. Sometimes you'll see in a diagram little coils holding these in place, and those are not shown for clarity sometimes, but those are also another protein called titin. And so what happens in a sarcomere is that when they're allowed to, the myosin thick filaments will grab onto the actin and they will pull them toward the middle, which shortens the sarcomere. And if this were an entire muscle, it's going to be shortening the muscle, making it contract. So we're going to get into the specifics of how that happens and what causes it, what makes it trigger, and what makes it stop. So now we're going to talk about what triggers those events to happen. Now at rest, the part of the actin that lets the myosin bind to it is actually covered up. And it's covered up by a long, thin protein called tropomyosin and little globular proteins called troponin. Now, when the muscle needs to contract, the tropomyosin needs to be pushed away from the myosin binding sites that are on the actin. So what happens is calcium ions, tiny calcium ions, bind to the troponin, and it changes its shape in a way that pushes the tropomyosin off of the myosin binding sites of the actin. And as soon as the tropomyosin is moved away, then the myosin can bind to those and pull it. And it would begin something called the contraction cycle, which we'll go over in a moment. When that muscle fiber needs to relax again, the calcium is removed from that space, the troponin changes back to its original shape. The tropomyosin covers the binding sites on the actin, so the myosin can no longer grab onto it. So next we'll talk about what triggers the calcium to be released in the first place and what triggers it to be reabsorbed. And then we'll end with the contraction cycle. So I just mentioned that calcium ions bind to the troponin and move the tropomyosin away from the binding sites on the actin. So the question now is, what causes the calcium to be released? So what I've drawn here are our actin and myosin filaments of a sarcomere inside of a muscle cell. The inside of a muscle cell is called the sarcoplasm. The surface of a muscle cell, this is represented here. So this would be an entire muscle cell, a muscle fiber. And the surface, instead of being called a plasma membrane, is called the sarcolemma. The sarcolemma, the surface of the cell, has parts that go deep into the middle of the cell. They're called transverse tubules, or just T-tubules. They're transverse because they are at right angles to the surface of the cell. The T-tubules go down and they're connected to these bags or sacs inside the muscle cell. Now those originally were smooth endoplasmic reticulum, but they've been repurposed in a muscle cell to hold calcium ion. And instead of smooth endoplasmic reticulum, they're now called sarcoplasmic reticulum. And then there would be some type of neuron, a motor neuron, connected to the surface of the cell at a place called the motor end plate. So what happens is a nerve impulse, an electrical signal comes down the neuron. It releases neurotransmitters, things like acetylcholine, and that causes an electric signal to start on the sarcolemma. The electric signal spreads out, and the electric signal would go down the transverse tubules and trigger the sarcoplasmic reticulum to release calcium ions into the sarcolemma. And it's those calcium ions that are going to bind to the troponin, which will move the tropomyosin off of the binding sites so that the myosin can grab onto the actin and pull it, make the muscle contract. When the muscle is ready to relax, the calcium ions are reabsorbed back into the sarcoplasmic reticulum. The troponin changes shape to its original shape. The tropomyosin will again cover the binding sites on the actin filaments. So now we're moving to something called the contraction cycle. And this is what happens when the troponin has changed shape because of the calcium, when the tropomyosin is moved out of the way. This is really just the interaction between the actin and the myosin now. So when the binding sites are exposed, the myosin will bind to the actin. The head of the myosin binds to the actin filament. What happens next is that this head that swivels bends, and so it pulls the entire actin filament this way. At this point, a chemical, a low energy chemical called ADP, is released. Now these two things, the myosin and the actin, are going to be bound together and stuck together until a high energy molecule called ATP comes in and binds to the myosin, and then they can be released. But the head is still in that bent position. So next, the ATP molecule, that high energy molecule, is split into ADP, a low energy molecule, and a phosphate group, and that energy is used to make it go from the bent position to its starting position. So the energy is used to go from here to here, and then we end back up here, where it can grab further down on the actin filament and bend, and then let go, and then reset, and then grab and bend and let go and reset, keeping the direction of the actin filament, the thin filament, keep pulling it in this direction. And again, this cycle would continue until the calcium is removed, the troponin changes shape, the tropomyosin covers up the actin so that the myosin cannot bind to it. So those are the basics of how a muscle contracts. Hopefully it was clear enough. Thanks for watching everything. And if you still have questions after watching this video, make sure you ask those questions to your instructor to get them answered. We'll see you in class.
Boop, 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 boop.